Imagine if a doctor could predict a disease before symptoms appear. Imagine discovering a new medicine without testing millions of chemicals in a laboratory. Or imagine understanding the complete genetic blueprint of a human being using a computer. Sounds like science fiction, right? Well, this is exactly what a fascinating field called bioinformatics helps us to do. Today we are going to understand bioinformatics in the simplest possible way, even if you have never studied biology, computer science or genetics before. So, what is bioinformatics? Bioinformatics is simply using computers and technology to understand biological information. Let's think in a way. Every living organism, from bacteria to plants to humans, contain an enormous amount of biological data, that is DNA sequences, RNA sequences or protein sequences. The problem is that this data is far too large for humans to analyze manually. That's where computers required. Bioinformatics combines biology, mathematics, statistics and computer science to make sense of biological data. So why do we need bioinformatics? Let's take an example. The human genome, the complete set of human DNA, contains about 3 billion letters in the form of A, T, G, C. If you tried reading it like a book, it would take years. Scientists needed powerful computers to store, organize and analyze this information. Bioinformatics help us answer the questions like which gene cause a particular disease, how do viruses evolve, which medicines will work best for a patient, how can we improve crop production. So without bioinformatics, analyzing such huge amounts of biological data would be nearly impossible. Bioinformatics means biological, and informatics means information. The term coined by Dutch scientists Pauline Hogbach and Ben Hasper in 1970. 80% that's how similar our protein coding sequence or gene are those of mice. Humans are larger, smarter and live more than the mice, but humans do not have fur and tail. The reason we have been able to calculate the similarity to mice due to the massive efforts to the sequence the genome of humans and an undertaking called the Human Genome Project. It includes the sequencing of genome of mice, human and other organisms also. Genome sequencing involves reading the ATGC content that make up the DNA. DNA gives us the lot of information about what are genes and how they are organized in the human or in any organism and how even we treat the human disease. Genome sequencing involves generating very large sets of data. So we need powerful tools for it. This is where rapidly growing field bioinformatics concerned. It required extremely powerful computers are being used to store and manipulate all of the data, and people behind the computer are called bioinformaticians. How does bioinformatics help us to understand the similarity between the genes of two organisms? Looking for small making sequences, genome that match the other genome. This is done by word search with the help of computer by using bioinformatic tools. Computational tools and statistical techniques to analyze biological data such as DNA, RNA and protein sequences. It is a fast evolving interdisciplinary field that sits at the intersection of biology, computer science, mathematics and statistics. According to the National Institute of Health, bioinformatics is defined as research, development or application of computational tools and approaches for expanding the use of biological, medical, behavioral or health data, including those to acquire, store, organize, archive, analyze or visualize such data. According to National Center for Biotechnology Information (NCBI), bioinformatics is defined as the field of science in which biology, computer science, and information technology merge to form a single discipline. According to European Bioinformatics Institute (EMBL), bioinformatics is defined as the application of computer technology to the management and analysis of biological data. This emphasizes data handling and computational analysis. According to NCBI, bioinformatics has three main components. First, the development of algorithms and statistics, application of computational tools, and the last one is development of biological databases. These components help us to understand how the field actually works in practice. The first component is the development of algorithms and statistics. This means scientists design mathematical model and computational method to study biological data. For example, when we want to compare DNA sequences of different organisms, algorithms help us to find similarities and differences. Stats are used to make sure the results are accurate and meaningful. The second component is the application of computational tools. Once the tools are developed, they are used to analyze and interpret biological data. These tools help in studying nucleotide sequences, which are DNA and RNA, as well as amino acid sequences in protein. Through this, scientists can predict protein function, understand gene expression, and study mutations. The third component is the development of databases. Since biological data is extremely large, it must be stored properly and accurately. Bioinformatics create databases that allow efficient storage, easy access, and proper management of this data. These databases are very important because researchers around the world can access and share information easily. Bioinformatics has three major branches. These are Genomics, Transcriptomics, and Proteomics. Genomics is a study of whole genomes using computational and statistical tools to store, analyze, and interpret large-scale DNA sequence data. Transcriptomics is a study of the complete set of RNA transcripts in a cell or an organism, analyzed using computational tools to understand gene expression patterns. Proteomics is a study of complete set of proteins or proteomes in a cell or organism using computational tools to analyze their structure, function, and interactions. Now let's look at the scope of the bioinformatics. First, genomics. In genomics, bioinformatics helps us to study the DNA sequences. It allows scientists to identify genes, understand mutations, and even compare genomes of different organisms. This has been crucial in projects like the Human Genome project, which mapped the entire human DNA. Transcriptomics is a study of transcriptome, that is the complete set of RNA molecules produced in a cell or organism at a given time. It helps us in studying gene expression patterns. One of the most important scope of transcriptomics is understanding gene expression patterns. One of the most important scope of transcriptomics is understanding gene expression patterns. In simple terms, gene expressions tell us which genes are turned on or off in a particular cell at a specific time. For example, a liver cell and a brain cell contain the same DNA, but they function differently because different genes are expressed. Transcriptomics help us to identify these differences. By studying gene expression patterns, scientists can understand cell function and specialization, identify disease-related genes, and they can compare healthy versus diseased tissue. And it also helps us to study how organisms respond to stress, drug, or environmental changes. Another major area in transcriptomics is RNA sequences, which help us to identify all RNA molecule present in a sample. We can measure the quantity of gene expression. We can detect new transcripts and alternative splicing events. In proteomics, we study proteins, the functional molecules in our body. Bioinformatics helps us to predict the protein structure and function, which is essential because proteins control almost all biological processes. It also helps us to tell the protein-protein interaction networks. Bioinformatics plays a huge role in identifying drug targets and designing new medicines. Instead of traditional trial and error methods, scientists now use computer simulations to predict how a drug will interact with a target molecule. It will save both time and cost. Personalized medicine is the patient-specific treatment strategies. It is the future of healthcare. Bioinformatics allows doctor to design treatments based on the individual's genetic makeup. So instead of one-size-fits-all, treatments become more effective and tailored to each patient. It comes under the pharmacogenomics. By using personalized medicine, we can detect or we can predict the genetic disease pattern in an organism. Another important area is agriculture. Bioinformatics helps to improve the quality of crop increase the yield, and develop disease-resistant plants. This is especially important for countries like India, where agriculture plays a vital role in economy. It also helped to raise the genetically modified organism and in livestock enhancement. Then we have data science in biology. Modern biology generates huge data sets, and bioinformatics uses artificial intelligence and machine learning to analyze them. This helps in predicting disease, understanding patterns, and making scientific discoveries faster. Next is systematic biology or system biology. It is the studying complex biological systems or network analysis of genes and proteins. Instead of studying one gene or protein at a time, bioinformatics help us to understand how entire biological systems work together, like how cells function as a whole. Now look at the applications of bioinformatics in different fields, mainly medicines, agriculture, and research. Firstly, we will look for application in medicine. Over the past few decades, bioinformatics has not just supported healthcare, it has transformed it completely. From understanding disease at the genetic level to designing drugs and vaccines. Over the past few decades, bioinformatics has not just supported the healthcare, it has transformed it completely. From understanding the disease at the genetic level to designing the drugs and vaccines. Bioinformatics plays a crucial role in modern medicine. The major applications are here. First one is disease gene identification. At the core of many diseases, especially chronic and genetic disorder, are changes or mutations in genes. Identify these genes is crucial for understanding how a disease develops. In past years, finding disease-related genes was a slow and complex process, but now with bioinformatics tools and large biological databases, scientists can compare DNA sequences across individuals and can identify the mutations linked to disease and can analyze the gene expression patterns. For example, in cancer, bioinformatics helps identify oncogenes, genes that promote cancer, and tumor suppressor genes. In genetic disorder like sickle cell anemia, a single mutation can be traced and studied efficiently. Now move to the next application in medicine, that is drug discovery. Traditionally, developing a new drug is a very time-consuming process. It may include 10 to 15 years. It is extremely expensive and it is high risk with many failures. But nowadays, bioinformatics has revolutionized this process by introducing computational approaches. Instead of testing thousands of compounds in a lab, scientists can now identify drug targets, that is, specific proteins or genes involved in disease, and then use molecular modeling and simulation, and then perform virtual screening of compound. This means researchers can predict which molecules will bind to the target protein, how effective a drug might be, or what will be the possible side effects of that particular drug. Now the result is the drug development process becomes faster, costs are significantly reduced, and success rates are improved. Next, come to the personalized medicine. Traditionally, treatments are designed for the average patient, but in reality, every individual is genetically unique. Bioinformatics allows us to analyze a person's genetic makeup, the gene expression profile of that particular organism, and how the drug will respond in a particular organism. Based on this information, treatments can be customized. For example, some cancer therapies are designed specifically for patients with certain genetic mutations. Drug dosage can be adjusted based on how a patient metabolizes a drug, which leads to higher effectiveness of treatment, reduce the side effects, and better patient outcomes. So instead of one-size-fits-all, medicines become patient-specific and precise. Last one is vaccine development. Developing vaccines traditionally required years of research and testing, but bioinformatics has dramatically improved this process. By using computational tools, scientists can analyze the genetic sequence of pathogens, that is disease-causing organism. They can identify antigenic proteins, that is a part of the pathogen that trigger the immune response, and can predict which components will be the most effective in the vaccine. This approach is often called reverse vaccinology. A very relevant example is the COVID-19 pandemic, where bioinformatics played a critical role in rapid sequencing of the virus genome identification. Now let's look at one of the most important application of bioinformatics, that is agriculture. As we know that populations grows day by day and their demand of food will be increases day by day. So farmers face a major challenge for this. They have to producing more food while dealing with limited land, climate change, pest and plant diseases. Bioinformatics is helping scientists address these challenges in smarter and faster ways. First, crop improvement. Bioinformatics allows researchers to study the genetic makeup of different crop varieties. By comparing their DNA, scientists can identify genes responsible for desirable traits such as better nutrition, drought tolerance, or faster growth. This helps in developing improved crop varieties more efficiently than traditional breeding methods. Next is disease resistance. Plant disease can cause significant losses in agriculture production. Using bioinformatics tools, scientists can identify genes that help plant resist pathogens such as fungi, bacteria, and viruses. These resistant traits can then be introduced into crops, reducing crop losses and decreasing the need for chemical pesticides. Another important application is genetic modification. Bioinformatics helps researchers to understand which genes control specific characteristics in plants or in animals. This knowledge enables precise genetic engineering, allows crops to become more resistant to pests, tolerate harsh environmental conditions, or provide enhanced nutritional value. Finally, the yield enhancement. By analyzing large amount of genetic and environmental data, bioinformatics helps scientists to identify factors which contribute towards. Next is yield enhancement. By analyzing large amount of genetic and environmental data, bioinformatics helps scientists to identify the factors which contribute towards high crop productivity. This leads to the development of high-yielding varieties that can produce more food from the same amount of farmland. In simple terms, bioinformatics is helping agriculture become more productive, sustainable, and resilient. It enables us to grow healthier crops, protect them from disease, and improve food security for a growing global population. Now we will explore an essential dimension of bioinformatics, that is its application in scientific research. If we think about modern biology, one thing becomes very clear. Research today is deeply data-driven. From DNA sequences to protein structures, we are generating massive amount of biological data every day. Managing and interpreting this data would be nearly impossible without bioinformatics. Bioinformatics acts as a bridge between biology, computer science, and stats. It enables the researchers to uncover meaningful insights from complex datasets. The three major research applications are evolutionary studies, molecular biological research, and data analysis in life sciences. First, talk about evolutionary studies. One of the fundamental questions in biology is, how did life evolve over time? Bioinformatics provide powerful tools to answer this question. By comparing DNA, RNA, or protein sequences across different organisms, scientists can identify similarities and differences and can trace the common ancestry patterns and construct phylogenetic trees which represent evolutionary relationship of one organism with the another. For example, humans share a large portion of their genes with other organisms like mice and even bacteria. By analyzing these similarities, we can understand how species diverged over time. Bioinformatics tools such as sequence alignment help researchers to detect conserved regions in gene and can identify mutation and evolutionary changes and study genetic variation across population. Now let's move to molecular biology research. Molecular biology focuses on understanding the structure and function of biological molecules such as DNA, RNA, and proteins. Bioinformatics plays a critical role here by providing tools to analyze gene sequences, predict protein structures, and study gene regulation and expression. For example, Researchers can identify coding regions in DNA and can predict how a protein will fold and function. And we can study interaction between different biomolecules. Another key area is functional genomics, where scientists aim to understand what genes actually do and how it perform a particular function. Another important aspect is protein structure prediction. Since protein function depends on its structure, bioinformatics tools help model 3D structures. Another important aspect is protein structure prediction. Since the protein function totally depends on its structure, bioinformatics tools help model 3D structures. This is crucial for understanding disease and designing drugs. Bioinformatics also help in studying gene regulation networks, signal transduction pathways, mutations and their effects on protein function. Now let's discuss the third and the most fundamental application, that is data analysis in life sciences. Modern biological research generates enormous data sets such as genome sequencing data, transcriptomics data, proteomics data. Analyzing this data manually is impossible. This is where bioinformatics becomes indispensable. Using computational tools and algorithms, researchers can store and organize large data sets in databases and can perform statistical analysis and can visualize complex biological information. For example, identifying patterns in gene expression data, detecting biomarkers for disease, clustering genes with similar function. To better understand the importance of bioinformatics in research, consider the large international projects like Human Genome Project relied heavily on bioinformatics for sequencing and analyzing the human genome. Today, almost every research lab uses bioinformatics tools for analyzing experimental data. From studying evolution to designing experiments, bioinformatics is involved at every stage of research. In wholesome, bioinformatics is the digital backbone of genetically modified organism development, transforming how scientists design, test, and monitor modified life forms. It bridges the gap between new genetic data and functional and stable organism. Today we are going to study the history and evolution of bioinformatics, an important topic that explains how biology and computer science came together to create one of the most powerful scientific fields in modern research. Before understanding modern bioinformatics, we must first understand its origin. The early beginnings of bioinformatics started even before computers existed. At that time, scientists were already trying to understand biological information manually. Researchers compared protein sequences, DNA patterns, and evolutionary relationships using paper-based methods and mathematical calculations. This process was extremely slow and difficult because biological data was increasingly rapidly. Imagine comparing thousands of DNA letters manually without computers. It required huge effort, time, and accuracy. During the 1950s and 1960s, molecular biology started rapid growth patterns. One of the biggest milestones occurred in 1953 when two scientists, James Watson and Francis Crick, discovered the double helix structure of DNA. This discovery completely transformed biological sciences because scientists finally understood how genetic information is stored and transferred in living organisms. DNA became recognized as a blueprint of life. At this stage, scientists realized that biological information could be studied systematically. However, they also understood that future research would generate enormous amounts of data that would eventually require computational support. So even though the term bioinformatics did not officially exist yet, the foundation of bioinformatics had already begun through the sequence comparison, molecular biology, and evolutionary studies. This period can therefore be considered the conceptual birth of bioinformatics. During the 1960s and 1970s, computers started being used in biological research for the first time. This was revolutionary because scientists were now able to store and analyze biological information electronically. Instead of manually. One major contribution came from the National Biomedical Research Foundation, also called NBRF. The first comprehensive collection of amino acid sequences was compiled in a publication called the Atlas of Protein Sequence and Structure. This was important because scientists could now systematically organize protein information. A major personality in the era was Margaret Dayhoff, who is often called the mother of bioinformatics. Her contributions were extraordinary. She developed the first protein sequence databases. And methods for comparing sequences and the famous point accepted mutation substitution matrix. The PAM matrix helped scientists measure evolutionary changes between proteins. It became one of the earliest computational tools for sequence analysis. It explains to students that before Dayhoff's work, biological data was scattered and difficult to compare. Her databases created order and structure in biological information. This period marks the transition from simple biology to computational biology. Computers were no longer just machines for calculation, they became tools for understanding life itself. This stage also introduced the idea that biology could generate digital data, which later became the core of bioinformatics. As a computational biology developed, scientists realized that they needed organized systems to store rapidly increasing biological information. This led to the development of biological databases. During the 1970s and 1980s, biological data began increasing very rapidly. Scientists around the world were generating DNA sequences, protein sequences, and experimental results. Without proper organization, this information could easily become impossible to manage. Therefore, several important biological databases were developed. One important database was EMBL, established in Europe in 1980. Another important database was the DNA Data Bank of Japan, DDBJ. The Protein Information Resource or PIR was also developed by NBRF. These databases became central repositories for storing biological information. You can compare them to digital libraries. Just as libraries organize books, biological databases organize DNA sequences, protein structures, and genomic information. These databases allowed scientists worldwide to store information, retrieve data quickly, share discoveries, and compare biological sequences. This period introduced one of the most important principles of bioinformatics: data organization and accessibility. Without databases, modern genomics and bioinformatics would not be possible. Students should understand that databases are still the backbone of bioinformatics today. Modern databases contain billions of sequences and are accessed daily by researchers worldwide. Once databases were established, scientists need faster methods to generate DNA data. This led to one of the greatest revolutions in molecular biology, that is DNA sequencing. One of the most important developments in bioinformatics history was the invention of DNA sequencing. Frederick Sanger developed the Sanger sequencing method. This technique allowed scientists to determine the exact order of nucleotides in DNA. For the first time, researchers could read genetic information accurately. This was a historic breakthrough because of Sanger sequencing. Now, the result of Sanger sequencing was massive increase in sequence data. The genomic research accelerated and computational tools became absolutely necessary. Suddenly, scientists had more biological data than humans could analyze manually. This created a strong need for computational analysis, sequence comparison tools, and automated algorithms. As a result, sequence alignment techniques began developing rapidly. Sequence alignment helps compare DNA or protein sequences to identify similarities, mutations, evolutionary relationships, and functional regions. This was the beginning of large-scale computational sequence analysis. Students should understand that sequencing technologies transformed biology from a small data science into a big data science. After sequencing technologies expanded, the 1980s became a decade of rapid computational tool development. The 1980s represented a major expansion phase in bioinformatics. During this decade, scientists developed many computational tools to analyze biological sequences efficiently. One important development was the FASTA algorithm. FASTA allowed rapid comparison of biological sequences. Previously, sequence comparison was extremely slow. FASTA significantly improved speed and accuracy. Later, many sequence alignment programs were developed. These tools help scientists to identify genes, compare proteins, study evolution, and predict the biological functions. At the same time, computer systems themselves became more powerful. The improved computing technology allowed researchers to process larger biological datasets. Bioinformatics application expanded into gene identification, protein analysis, and evolutionary biology. This decade proved that computers were becoming essential in biological sciences. Students should recognize that modern bioinformatics software evolved from these early computational tools. Today's advanced genomic software, AI system, and protein prediction tools all originated from these foundational developments. As a computational biology continued progressing, scientists launched one of the largest scientific projects in human history, that is the Human Genome Project. The Human Genome Project was one of the most ambitious scientific projects were ever conducted. It started in 1990 and was completed in 2003. This was an international scientific collaboration involving researchers from multiple countries. The main goal was simple but enormous: to sequence the entire human genome. That means scientists wanted to identify all human genes and determine the complete DNA sequence of humans. The project generated an enormous amount of biological data. Without bioinformatics, this project would have been impossible. Bioinformatic tools were required for storing genomic data, assembling DNA fragments, sequence analysis, and genome annotation. Students should understand that the Human Genome Project transformed medicine and biology permanently. Its achievements included the identifying disease-related genes, understanding human evolution, improving diagnostics, and opening the field of personalized medicine. The project also accelerated the development of high-performance computing, genomic databases, and advanced algorithms. Today, modern genomics, transcriptomics, and precision medicine all depend heavily on the foundation created during the Human Genome Project. Today, modern genomics, transcriptomics, and precision medicine all depend heavily on the foundations created during the Human Genome Project. At the same time, another powerful technology helped spread biological information globally. During the 1990s, the rise of internet dramatically changed bioinformatics. Scientists could now share biological information globally within seconds. Several important online resources were introduced. One major resource was National Center for Biotechnology Information, or NCBI. NCBI became one of the world's largest biological information centers. Another major tool was BLAST. BLAST stands for Basic Local Alignment Search Tool. It allows scientists to compare DNA or protein sequences rapidly against massive databases. Other important databases included Swiss-Prot for protein information and Protein Data Bank for protein structures. The internet created several benefits, that is, global data sharing, faster scientific communication, and easy access to genomic information. Previously, researchers depended on printed journals and physical data exchange. Now, scientists worldwide could instantly access the same biological data. This increased collaboration and accelerated discoveries tremendously. Students should understand that modern bioinformatics heavily depends on internet-based databases and online tools. As technology improved further, bioinformatics entered the modern era with advanced sequencing, artificial intelligence, and big data analysis. In the 21st century, bioinformatics entered a highly advanced technological era. Several powerful technologies transformed the field. One major innovation was next-generation sequencing. Next-generation sequencing allows rapid sequencing of millions of DNA fragments simultaneously. If we compare it to Sanger sequencing, the next-generation sequences is faster, cheaper, and capable of generating enormous datasets. Modern bioinformatics also uses artificial intelligence, machine learning, cloud computing, and big data analytics. Artificial intelligence and machine learning help researchers to predict protein structures, identify disease patterns, and analyze complex biological datasets. Cloud computing allows scientists to store and process massive genomic data online. Big data analytics helps interpret extremely large biological datasets generated by modern sequencing technologies. Today, bioinformatics is applied in healthcare, agriculture, drug discovery, forensic science, and personalized medicines. Now let us look toward the future and understand where bioinformatics is heading next. The future of bioinformatics is extremely promising. Emerging technologies are expected to revolutionize healthcare and biological research even further. One of the major future trends is artificial intelligence driven healthcare. Artificial intelligence will help doctors to diagnose the diseases faster and more accurately. Another important area is precision medicine. Precision medicine focuses on patient specific treatment based on individual genetic information. Genome editing technologies like CRISPR are also transforming modern biology. CRISPR allows scientists to edit genes with remarkable precision. Bioinformatics is essential for analyzing and designing these genetic modifications safely. Another future direction is multi-omics integration. This means combining the genomics, transcriptomics, proteomics, and metabolics data together. By integrating multiple biological data sets, scientists can better understand complex diseases and biological systems. Further, bioinformatics will also support personalized treatment planning where therapies are customized according to a patient's genome. Now let us summarize the major milestones in the evolution of bioinformatics. In 1953, Watson and Crick discovered DNA structure. During the 1960s, computers entered biological research. In 1970s, biological databases were developed. In 1977, Sanger sequencing revolutionized DNA analysis. In 1980, sequence alignment tools were emerged. From 1990 to 2003, the Human Genome Project transformed genomics. In 2000, next generation sequencing expanded rapidly. Today, AI-driven bioinformatics and big data analysis dominate the field. This timeline shows how bioinformatics evolved from simple sequence comparison into one of the most advanced scientific disciplines in the world. As I told you earlier about important biological databases, now I will show you how will they look like. First of all, I am going for EMBL. First of all, I will go for EMBL, that is European Molecular Biology Laboratory, and EMBL has a as I told you that rise of internet and online databases will help us to access online resources like BLAST, FASTA, NCBI, many more things we can access online. Now to know about the sequence of anything and to show how they look like, we want everything in a FASTA format. We will retrieve our nucleotide sequences or protein sequences in FASTA format. So how it will look like, I will show you that. The result for P53 protein will take about mus musculus FASTA format, right? Okay, now you can check it. As you know that it is a sequence of protein, so the alphabet will be MTA, means name of the amino acids. Okay, there are 20 amino acids and they will be in single alphabet format. You can check here M for methionine, likewise. So this is the FASTA format of protein look like. Now if I'll check how the nucleotide or genes FASTA format will look like, I will show you that also. If I check for this nucleotide only, Homo sapiens, we will check about Homo sapiens. Yes, this is Homo sapiens. P53 genes look like. Now we can check that DNA is consist of 4 nucleotides only. That's why here ATGC content will be here. The repetition of these 4 letters only. This is how FASTA format of gene and protein will look like. We will retrieve our data from here, from this site. Now we'll check how BLAST tool will look like. If I check it, it will be look like in this manner. It will look like BLAST stand for Basic Local Alignment Search Tool. Now we can check here. We can do nucleotide BLAST. We can do protein BLAST. Okay, nucleotide BLAST for this. This is mainly for DNA and RNA sequences. Now I will go for. It will give you this page. Here the modification and variety types of BLAST. BLAST and for search a nucleotide databases using a nucleotide query. BLAST P for protein databases for protein query. BLAST SX for protein databases using a translated nucleotide query. And TBLAST N for search translated nucleotide database using a protein query. And TBLAST X is for translated nucleotide database using a translated nucleotide query. So it is how these variants will work. Now for this we have to enter query sequence. We have to enter our query. It will be known or unknown sequence. We can fetch it from FASTA sequence in FASTA format only, and we can fetch it from NCBI website as I show you earlier. Then you can write what will be the job title over here, what will you want to perform. If you want to perform nucleotide BLAST, you can click here. If you want to perform protein BLAST, you can click here. And these are the variants. And whatever you want to do, just put the title here and do the BLAST. This is how the BLAST page look like.